Hello, and welcome to this US 275 Scientific Ethics lecture, where we're going to start to look into the history of human subjects research. And so basically, where we're at in the course right now is that we're going to make a little bit of a shift from looking at different ethical models to kind of the application of those ethics and those ethical ideas and morality to various aspects of scientific research. And the first set we're going to look at is going to be human subject research and take a look at it in relationship to what we've been able to learn about the body through research and improvement for the quality of life of individuals. So if we take a look at it just in a very basic sense, we can see that the life expectancy has increased pretty dramatically. Sorry about that, not quite sure what happened. Has increased pretty dramatically over the last 200 years or so. So if we look at it at about 1840, the average life expectancy was in the range of about 47, 48 upwards, and this is the most recent one I could find, 1978, showing a pretty linear line going up here to kind of the average life expectancy, in the late 70s is being about 76, 77. And so about a 30-year increase in life expectancy over a 200-year period. And it's, at least on this projection, projected to continue to rise. And it, probably is not going to continue to rise indefinitely. But, it is showing kind of an improvement in life expectancy and hopefully in quality of life. What we can see by the changes that have occurred within society is that in the first 100 years, from about 1840 to about 1940, 1960, we were looking at a decline in early and midlife mortality. Many cases, in the 1800s, infants would die, at childbirth or very soon after birth. And made it very difficult to survive a longer period of time. Since about 1950, 1960 on, what we've looked at is declining kind of late life mortality. And so learning how to deal with some of the, the disease processes that are more commonly observed in older individuals, things like cancers and heart disease and things like that. If we, take a public health perspective look at this and try to figure out what is associated with this pretty dramatic increase in life expectancy, we see that there are two primary areas. We had increases and advances in public health, sanitation, indoor plumbing, sewer systems, things like that. So getting waste materials kind of isolated and away from the populace. And we're also looking at kind of a richer, more complete diet, which has been established over the last 200 years. So at least within the United States, less undernourishment, less malnutrition. Malnutrition is basically not eating the right things. We still have a lot of work to do in this, and we're looking at this from the perspective of the United States and some of the developed world countries where we need to be able to expand this out to other regions as well. And so we've had advances in public health. We've also had advances within healthcare, development of medicines for the treatment of diseases, development of treatments, and as well as medicines for the prevention of diseases, which has really, again, shown a pretty dramatic increase in the life expectancy over the last 200 years or so. All of this comes about because we've done research, we've done science, and we've learned more about the body and how to improve conditions for people to be living in. If we take a look at this again from a historical perspective, we want to learn about the body, be able to understand it, and be able to, fix it when something goes wrong, to, set bones when they're broken, or, treat disease when an individual becomes sick. So we can take a look at the very basic process of studying human cadavers in an effort to try to learn about the human body. And we see that this is, kind of ebbed and flowed over the last, 2,000 years or so. It was very common, about 200 BCE for Greek physicians to, study, in many cases, dead gladiators, dead slaves. So we get some ethical issues associated with that. But basically, dissecting the body, looking at the different organs, how the organs are connected, and trying to figure out how the body worked. This, however, the dissection and studying of the human cadavers was banned in the Roman Empire, and for an extended period of time was felt to components of witchcraft and deviltry and devil worship and all kinds of things like that. So it was not in common practice during that period of time. But by the time the Renaissance period came, we saw that it had returned as a central aspect of the training of physicians, as well as doing research on the body. And we've seen that this has continued up through today, where it's often used for the training of clinicians, even for those students that are going into PA and PT. PT especially is going to have extensive cadaver dissection. PA has been doing that as well in the past, making a transition to some of the newer technologies that are available for studying the body without the use of cadavers. So, the principle is still there, but we're not necessarily using dead human bodies to be able to do that. More recently, kind of taking a look at kind of an example of scientific research to try to come up with a treatment or a prevention for the disease. In the late 1800s, we take a look at Edward Jenner. Edward Jenner was a British physician, an English doctor, in about the 1700s now. And he was looking at smallpox. Smallpox is a very deadly virus that is fairly infectious, and if you get it, at least 200, 300 years ago, it was a situation where we didn't have the medicines, we didn't have the treatments to be able to help the person get well, and therefore it became very deadly. But Jenner observed that dairy maids, kind of like the example over here, a woman that goes through, works with dairy cows, often milking them, multiple times a day, didn't seem to get the smallpox virus. And so when he, started to take a look at these dairy maids, what he observed is that the dairy maids were exposed to a version of cowpox. Cowpox, like smallpox, is a virus. But cowpox is a milder virus. It doesn't cause, massive disease and death, but it, will cause a little bit of, sickness associated with it. It can be contracted by the milkmaids because they're, milking the cows. So, they're coming into direct contact with the cow's udders, multiple times a day. So they're going to be exposed to and ultimately get this cowpox. And it seemed as if exposure to the cowpox virus gave them some type of immunity to smallpox. And Jenner, being a scientist, or at least applying the scientific method, thought that this is important to look at as a possible way of preventing this very devastating disease. And so what Jenner did, I can advance my slides. What Jenner did is he basically, went out, found a dairy maid that had some cowpox lesions on their skin. And so you think about, almost like a chickenpox virus. When you have chickenpox, you get the little sores on your skin. They'll scab over as they're healing. And what Jenner did is basically kind of collect some of those scabs and collect some of the tissue kind of underneath them. And again, a little bit of a bit of unethical treatment, especially from our thoughts today. He basically took that sample of the dairy maid hand lesion, the lesions that were exposed to the cowpox, injected them unknowingly into an 8-year-old boy who didn't know it. Jenner knew what was going on, but the 8-year-old boy didn't. So he injected potentially this disease-causing thing, cowpox virus potentially, into this child. The child developed fever, lost an appetite, felt discomfort, but ultimately recovered. And so basically he was able to show that these lesions, almost like the scabs that he took from the dairy maid, were infectious and could cause the disease. So that was interesting in and of itself. But then Jenner actually did the same thing with smallpox virus. And again, incredibly unethical and hopefully would not even be considered to be done now at this point in time. But Jenner took the smallpox virus, injected it into this child that had gotten cowpox and recovered. Normally, it would have been pretty likely that this child would have died as a result of this. But the kid didn't get sick. He had developed a resistance to getting that disease. He basically developed immunity to this. And so this is a good example of, or a good first example of the concept of immunization, to kind of expose individuals, in this case, to a milder version of the virus, in this case, a related cowpox virus. And the body's reaction to this cowpox virus made it so that it was resistant to the smallpox virus when it was exposed to it later. And so it's almost like a vaccine created by the cowpox virus provided immunity against the deadly smallpox. Now, taking a look at other examples, and again, taking a look at this from a historical perspective, in World War II, and many of us have heard these stories, the Nazi scientists, Nazi doctors, did thousands upon thousands of experiments on prisoners, on the people that they basically felt were not in line with their goal for a master race. So, taking a look at Jewish people, people that were homosexual, people that were gypsies, basically people that didn't fit with the German mindset on what was a good German kind of they called it stock, but good German body type and personality and all that. And so he did all kinds of these different experiments on them in many, many unethical ways. And we'll have a follow-up on that as well. They'd often do twin experiments. So they would take one child, one identical twin, so ideally genetically identical to one another, and expose them to really awful conditions to be able to understand the disease process, and then look at the other twin and try to understand it. And so, what the Nazi scientists were doing was essentially a form of eugenics. They were trying to learn about the body in a way so that they could improve the human population by promoting certain genetic groups, genetic characteristics, the Aryan stock, if you want to think about it that way, the good German kind of characteristics, but excluding others. So eliminating, other people from the society that didn't fit with their ideal. And this was what they described as an effort to make a more perfect race. But in general, they were doing incredibly horrific and unimaginable experimentation on prisoners and on people that had no way to, defend themselves or, use their autonomy to say they didn't want to be, involved in this. And so again, huge ethical concerns with this. And it didn't just happen in Germany. If we take a look at what happened in the Asian and Pacific theater during World War II, the same type of thing happened in the Japanese army. They had a Unit 731 that conducted biological and chemical warfare research in China during World War II. And they would basically go in and test biological weapons, and in some cases chemical weapons, and basically go through and release them into the population, into the general population. So not using them on soldiers as an act of war, but using them against civilians. And so what we saw, depending upon where the scientists and the people from Unit 731, the Soviets moved into the region, captured a lot of them. The United States did as well. The Soviet forces held the Khabarovsk war crime trials and convicted a lot of the people that were involved with these studies. In contrast, Douglas MacArthur took these people, the prisoners that were captured, or the scientists that became prisoners at the end of the war, and basically granted many of them immunity and used them to kind of jumpstart the US biological warfare program. Just in terms of keeping things complete, to go back to Nazi Germany, there were the Nuremberg trials that happened after World War II that not addressed, because it's impossible to address some of the atrocities that occurred, but basically held people accountable for what was going on in Nazi Germany. And in many cases, it was soldiers, but in other cases, it was the scientists doing the experimentation as well. And so the Nuremberg trials were used to get a hold and to respond to some of the atrocities that happened in Nazi Germany. Now, we looked at it in Germany, we looked at it in Japan, but, people in glass houses shouldn't throw, stones. This also happened in the United States. A good example of this is the Tuskegee syphilis study. This was a study that was launched by the Public Health Service before World War II. It was launched in about 1932. Full name was the Tuskegee Study of Untreated Syphilis in the Negro Male. But basically, it was another example of an incredibly unethical set of experiments that were done in an effort to try to understand about the disease progression, in this case, the progression of the disease syphilis. And so, again, these people that were subject in these studies were not informed about what was going on. They were told that they were going to be treated for bad blood, but no real explanation about what that bad blood was. But in many cases, there were individuals that developed syphilis and basically went untreated for a very extended period of time. Even though by the end of World War II, by about 1947, penicillin, the antibiotic penicillin, was a preferred treatment for syphilis. This wasn't given to the people in this study so that they could look at the full disease progression, the later stages of this very debilitating disease. Ultimately, the study lasted for 40 years. These people went, 40 years without treatment, and it resulted in huge disabilities and many deaths associated with it. And it only ended in 1972. Once that information started getting out. There was an investigative journalist that started looking into it, wrote some articles that generated lawsuits and a huge public outcry associated with it. Again, these things are incredibly unethical and hopefully never would be considered being done again, but there are instances, and it wasn't just an isolated incident. Another example of this was the Guatemala syphilis experiment. It was done kind of in concert with the U.S. Public Health Service, an NGO, the Pan American Sanitary Bureau, as well as the Guatemalan government, where they actually went in and intentionally infected a large number of Guatemalan prisoners, sex workers, soldiers, and mental health patients with sexually transmitted diseases, so that they could look at how the disease kind of spread through the body and the progression of the disease as it continued to kind of develop in an untreated way throughout these individuals. And again, another huge example of an unethical study that was done in the US past. In the Guatemala syphilis study, at least 83 individuals died, and probably more people died after that. More recently, within the US prison system, again, experiments done on humans, but done on individuals that really didn't have the ability to consent to participate in this. They didn't have any ability to, protect themselves or opt out or, basically, have any rights at all. And some really strange experiments were done within the US prison system. Transplantation of organs, in some cases sex organs from rams, goats, boars, executed prisoners. And this is a fake photo on the right-hand side of this. But there were efforts to try to, look at kind of combining kind of animal parts and human parts into hybrids, almost like a chimera. They did studies on tuberculosis, again, a very debilitating and devastating respiratory disease, incredibly infectious. Again, looking at that and the disease progression and how it spreads within a confined community. Did experiments on malaria as well. So lots of examples, again, on individuals that did not have the ability to consent to participate in the studies, and they had no protection really as a subject within these studies. This continued up into about the 1960s, where about 90% of the pharmaceutical research was done on prison inmates. And so, about 90% of the drugs that were being tested for development and use on the population. Again, the idea is that you want to make sure that they're safe and they're effective and they're not going to cause more problems. So you need to do human studies on them. And we hear a lot about clinical research now when we're talking about clinical trials for vaccines. But this, was done very extensively within the U.S. prison system, and it ultimately ended in the 1970s. And we'll talk in the next set of lectures about some of the things that came about to end these prison experimentation. But, again, unethical studies done on vulnerable subjects, which had no ability to, have rights or have ability to, protect themselves from what is going on. To put this into perspective, we said that up to the 1960s, about 90% of the pharmaceutical research was done on prison inmates. If we take a look at drug testing now, much of it is done overseas. And so from 2008 to 2013, there were over 58,000 experimental drug trials involving humans done in 173 countries around the world. And that was about a 2,000% increase. So, 20 times what it was, or I'm doing the math right, 20 times what it was in 1990, less than 20 years earlier. So there's been this huge movement of clinical research and experimental drug trials away from the United States, where potentially we have more rules and regulations, into other areas of the world where potentially subjects now are vulnerable. They don't have necessarily the same protections that they would have within the United States. If we take a look at that, by 2017, about 90% of the new drugs that were approved by the Food and Drug Administration, so about 90% of the drugs had been tested outside the United States and Canada. Now, this isn't saying that they were done exclusively outside of the United States, but there was a significant component of the clinical research testing the safety and effectiveness of this drug done outside of the United States and on populations that were potentially vulnerable. So that kind of gives a kind of a historical perspective, kind of highlighting some problems with how research was done kind of previous to the 1970s. In the next set of lectures, we're going to talk about some of the changes that came about in the 70s and some of the modifications to those rules that have occurred, over the last 50 years or so, to ideally protect research subjects and ensure that research is done in an ethical way. I've got a couple of readings for you today. We've got the historical and ethical and legal background of human subject research, which I think is a really good article to take a look at, and you can get that as a full text PDF. A question, is it ethical to use data from Nazi medical experiments from the conversation in about 2015? And then a very interesting dilemma. The surgeon had a dilemma using a Nazi medical test, the surgeon had a dilemma only a Nazi medical test could resolve. Was it ethical to use? And again, an article on that. And I think I'm going to include a video I recorded for another course that goes into a lot more detail about this specific situation. And so, that's going to be our second video for today. So I'm double using it because I used it in another course, but I think it's really relevant to what we're focusing in on today. As always, if you have any questions, feel free to email me. Thank you and have a great day.